Welcome to the MGH brain-PBM clinic rounds. My name is Paolo Cassano. Today is November 15, 2024, and it is my great pleasure to invite Dr. and Professor Saša Vlahinić, faculty at the Faculty of Engineering in Rijeka in Croatia. And Saša, please take it away and welcome today.
Thank you. Thank you for the invitation to present our work and for the possibility to be here with you. I will share my presentation. Okay, I hope everything is you can see.
That's great.
Yeah, okay, great. So, as you said, I'm coming from University of Rijeka, Faculty of Engineering. So we are engineers. This is our small group of researchers. We are doing EEG signal analysis, and we are also doing some research in assistive technologies. In more details, we have been done some research in brain-computer interfaces, BCIs, and actually today was the anniversary. So we are established as a faculty 64 years ago. Yeah. Okay, today I will be talking about our case study that we did a couple of years ago. I will explain the study, how we did it, and what kind of analysis we did in this study. We did a power spectrum analysis and then functional connectivity. And in the end, I will say something about what we are planning to do in the future, and in the meantime, what we have been done. Okay. So this was the study. The part it was a part of a project that started around 2020. And unfortunately, just before the COVID pandemic. So the idea was to do a bigger research with a lot of participants. In the end, we finished with one case because our target population were elderly people, and that was really, really tough at that time to work with this population. So we created this prototype to control diode clusters, and we had possibility to work at different frequencies, but since we didn't have possibility to try many things, we selected 10 Hertz as a frequency of stimulation with infrared light. We use the rectangular square wave. So what we did, the study lasted for 35 days, and we recorded EEG on the first day and on the last day. On both days, we did a recording before stimulation, 5 minutes eyes open, 5 minutes eyes closed. Then we did 10 minutes of stimulation, and then in the end, again, 5 minutes in both conditions. The subject was just in a resting state without particular task to do. In this way, we are hoping to see some short-term effects by comparing results before and after, and possibly also the long-term effect by comparing what has happened, what was the difference between the last day and the first day. Okay, the age of the subject at that time was 84 years, and with this self-assessment of his cognitive status, where the maximum score was 22 and 17 is the limit between the normal functioning, and 16 and 15 are indicators of mild to moderate cognitive impairments and early signs of dementia. Below that is a more severe indicator of more severe cases. I said it lasted for 34 days. For the first week, we did stimulation for 3 days, 3 times a week, and for the rest of the study, it was like 5 times a week. So first thing that we did, that we analyzed, was a power spectrum analysis. Why we did that? Because by studying the literature, we found that one consequence of cognitive problems and memory problems was this slowing of the EEG spectrum in general. So there is decrease in alpha spectrum and increase in theta band. Actually, those similar indicators occur in normal aging, but it is definitely more pronounced in case of problems. And also, it was proposed in the literature that this ratio between theta and alpha band is also a good indicator of cognitive problems. So we practically did analyze those 3 things: theta band, alpha band, and ratio between theta and alpha. So I will present now some results, and maybe this looks similar to neurofeedback. Actually, the original idea was to combine neurofeedback with PBM, but again, due to COVID and all the problems, we couldn't do both things, so we just focus on PBM. And yeah, as I said, stimulation was the 10 hertz. Probably it would be better if we tailor the frequency specifically to the subject, but we just use this 10 hertz just as standard alpha frequency in this case. So the goal was to increase the power in alpha band and decrease the power in theta band. And we did preliminary EEG analysis, and in fact, we observed that there was slowing on alpha band, and there was increase of theta band power. So we are hoping to achieve the opposite effect. As I said, to analyze what is the short-term effect, we did analysis comparing the results before and after for 1 day, and for the long-term effect, we compared the first day and the last day when we did recordings. Okay, so I will show some results. We used 16 electrodes for EEG analysis, and green dots indicate situation we had positive variations, what we wanted to achieve, while the red dots indicates electrodes where we didn't observe the positive effect. So this is the result for 1 day, short-term effects. And we can see that alpha power increased. It was more pronounced for the closed eyes than in open eyes conditions, but majority of the electrodes in the closed eyes had this positive variation. As far as long-term effect, we observed again better results for the closed eyes and more electrodes here are in green. That means that only 4 electrodes didn't have this. We didn't observe this effect. For the theta band, the results, short-term results, day 1 were predominantly positive. Most of the electrodes had a decrease of theta band power. So that was a good result. For the long term, not so good. Basically, it was half and half electrodes with and without positive results. Finally, as far as this spectrum analysis is concerned, this is the ratio, theta alpha ratio, and his variation. The best results for the spectrum analysis is for this theta alpha ratio that was predominantly positive for the most of the electrodes, 13 in this case for the first day, then little bit less but still good for the last day, after 35 days in total of this procedure. Long-term effect for theta alpha ratio were also good. 13 or 14 electrodes out of 16 did show positive improvement or decrease of this index. Okay, not only the power spectrum is considered when analyzing, trying to determine if there is a cognitive decline by EEG analysis. Also, functional connectivity is often considered, and we are trying to see if there is an improvement in information flow or connectivity between different brain regions. Of course, we used EEG, so we did functional connectivity using analyzing the same signals that we recorded. For functional connectivity, we can do both undirected and directed connectivity. In our case, we had a resting state, so we use undirected connectivity. Where we try to find stability in the phase difference between the two signals coming from different electrodes. And if there is stable phase difference, we then assume there is a connectivity between these electrodes. One big problem when we study functional connectivity is volume conduction, or actually we are seeing a signal coming from the same source from the brain on different electrodes. And that seems that these two electrodes are connected, but in reality, it's just an artifact. We are seeing the same signal. So on the left picture, we have a situation where we have this phase connectivity and practically zero phase lag, or 180. It can also be 180. And on the right hand side, generally we have a phase difference that can assume any different phases. So there are many measures and many indexes to evaluate functional connectivity. Some of them, as phase locking value, are susceptible to volume conduction, while the other ones are proposed to avoid, like phase lag index, weighted phase lag index. In this study, we used something that we proposed, and that is a complex correlation coefficient between two signals. And when we analyzed this coefficient, we can analyze different things. We find out that by analyzing absolute value of complex correlation, we have a similar behavior as a phase locking value. On the other hand, when we are analyzing imaginary part of this complex correlation, results are similar to weighted PLI. So we used imaginary part of complex correlation to see if there is on what is the state of connectivity before and after again. So these are the matrices that we get when we analyze functional connectivity. Actually, it's not from this study because there are many electrodes. We had only 16 electrodes, but more or less the same thing. So the color of each little rectangular is the value of represents the value of functional connectivity. For example, this little rectangular indicates connectivity value. Oh, wait, I can. Okay, so this value here indicates connectivity between O1 and PO7, if I'm not wrong. And it's a relatively high connectivity. We can see that bright yellow colors indicate good connectivity near 1, while dark blue colors indicates low connectivity near 0. In this case, these bigger rectangulars indicate volume conduction because we see a big group of electrodes that have high values, and this is PLV measure, so it's probably due to volume conduction. For example, this one measure that aims to avoid the problem of volume conduction doesn't have this problem. You cannot clearly see these big areas of high connectivity. Okay, so actually we get for each recording we get 16 by 16 values. So it was difficult to compare results before and after. So we used a global measure of efficiency coming from graph theory, and this measure practically calculates the path length between all nodes, and then calculates just one value. Again, we focused on alpha band to see if the connectivity in alpha band change after PBM. Okay, these are the results that I will try to explain. So on the left side, we have a situation before stimulation, and we don't see many connections. Actually, only connections that are that has value above certain thresholds are shown. So there are not many connections on the left side before stimulation, but after stimulation, we see there are much more connection. So this was short-term, before and after in 1 day, and similar results can be observed comparing the first and the last day. more connection and stronger connection after stimulation. So in this case, for the open eyes, we observed stronger effect and bigger difference, 5 times more after 35 days, while in the closed eyes that was less pronounced. So okay, of course, it would be much better to have a larger group of participants now. Then we can work without COVID. Probably it would be easier to do it. But I will say something about what we did. Meanwhile, we studied ways how to analyze dynamically connectivity, and I will say a few words about directed connectivity. So, what is dynamic functional connectivity? Statically, as I have shown before, we just get one number for the whole period of measurement, of recording. In this case, we are analyzing, for example, these 5 minutes with shorter narrow window of, I don't know, let's say 1 second, 10 seconds, or some other width of the window. And we are trying to see how this connectivity change dynamically when something is when we are recording this EEG. This could be used, for example, in brain-computer interfaces when we have some something occurs. So there is a question, and we expect an answer or some reaction in EEG signals. So we are trying to find a change in response, and it is then important to see how things change dynamically during the recording, and it is important to find when the change occurs. It will be also important to see how things dynamically change if the subject is doing a certain task, so we can observe what's going on. So on one hand, we would like to have a narrow window, but if we have too narrow window, then there is a problem of too much noise and large oscillations. So it's hard to find effective variations. If we take a wide window, then there is a problem that we lose in temporal resolution. So what we proposed, we tried to apply this algorithm based on intersections of confidence intervals rule. Basically is using statistical properties of the signals to establish the window width. Originally, it was used for signal denoising, and has different, since then it has been used for different things, and this was one possible application. Okay, so here we have some results. When we generated synthetic signal, because in that case, we exactly know what's going on, and so we have 3 intervals. The first interval is this one, when we have high connectivity, then we have a transition to low connectivity, and again, in the third one, again, we have a high connectivity. So what we want this fast transition, as we can see in this green line. We can see that this measure is nearly 1 on the first part, then there is a fast transition to 0, and then again it climbs more or less to 1 on the third part. But on this first and the third part, we see oscillation. There is no stable value. There is some noise in this estimation. On the other hand, wide window has more stability in this part, but we see that there is a slow transition from 1 to 0 and again from 0 to 1 on this second transition. So it's much harder to establish where exactly the change occurred. And our algorithm is this one, this purple color, I think. Okay, so it has, again, good narrow transition, so fast response time, and there are less oscillation when comparing to this narrow window. So in this case, the window width is adaptively changing, and the size of the window depends on the signal properties. Okay, so here are some numbers. Some results are better. So we have a better resolution, and there is some improvement in variability also, comparing to both a narrow window and large window. And finally, we are trying to extend this analysis also to analyze the direction of connection between the different regions, and to use measures for directed connectivity. In this case, what is often used is Granger causality that was proposed in econometrics, but it was used also for EEG analysis as well. Actually, causality may be a little bit misleading because it's not telling us if there is a causal relationship. Actually, it's telling us if knowing one signal can help us predict the other one. If knowing the other signal help us nothing in improvement of prediction, then there will be very low value of this causality, Granger causality. Okay, so the next things to do is to try to see how this measure can be applied both for the analysis of these results and maybe also in other fields. Okay, I think this is more or less everything. I hope it wasn't too technical, and I try to simplify as much as possible. So thank you for your attention.
Thank you, Sasha. Very, very intriguing. If you want to stop sharing, we can see the whole group, and I know there are more people than we see in the screen. So it's open to any questions that people might have. I had a question to begin with, and by the way, for everyone else, either speak up or raise your hand so that we can see you. I was wondering if in the entrainment, when you saw the increase in alpha, that was kind of converging over the 10 hertz that you were using, or not? Was it specific to the frequency you were pacing?
Good question. Actually, we focused on alpha band. And we also recording during this stimulation, but we found there are a lot of interference, so we couldn't use those signals. We used only before and after. Actually, I'm not sure.
Yeah.
But I think it wasn't specific to 10 hertz. It was more broadband.
Okay, great. Um, any other comments or questions?
And probably it would be better if we tailored the frequency of stimulation to the subject. Maybe to adapt it, there is a possibility, no problem with that, to adapt the frequency and to change it, and then maybe to have a bigger effect.
I see a question in the chat, whether this was just kind of a one subject experiment, or if there were more subjects, or if there's a plan for more.
The plan was to do more extensive research with many subjects and maybe try different things, not just one frequency. But as I said, we just couldn't do that at that time. Yeah, it would be good, but there some time has passed already. But yeah, maybe now will be the time to restart thinking about that again. Yeah.
Thank you. And also, still from the chat, is there any information that you might hypothesize to be useful from the dynamics of the alpha and theta band power over time?
Excuse me, I didn't understand.
Yeah, it's also in the chat. If there is any information that you might hypothesize to be useful from the alpha and theta band power over time. To a certain extent, you answer the question by showing the theta alpha ratio and how that summarizes the overall shift, and basically the increase in faster oscillations overall in the balance after those 3 times a week treatment for 35 days. I don't know if there's anything else. And Marty, if you are still on, you can comment if there's anything specific to your question.
I've got a general question.
Go ahead, Jens.
I'm curious to hear if whether or not there's any, I've just not noticed a study and I haven't looked. Has anybody proven that there is a correlation between pulse frequency and brain oscillations, and that if you increase the frequencies of pulse frequency from photobiomodulation, do you get a change in the oscillations in the brain that correlate with pulse frequencies? Anybody proven that yet?
I was also wondering if it's just DC, constant stimulation, would do the trick also. Yeah.
It's quite clear that pulse is in repair, and where there's certainly in the inflammatory phases that low frequency pulses, and 10 hertz is a good one, is better than 100 hertz. It's been shown in wound healing studies. It's been shown in the brain as well in I think it's a rabbit model of stroke, I think, with phototherapy. It's going back a long way. It's like 15 years ago. And I know other people have used frequencies on the brain, but I haven't seen comparison of different frequencies on the brain other than 10 hertz, 100 hertz in a stroke model. And brain isn't my area of expertise, but that just to be, I just like to see people comparing. I don't know what the frequencies, I don't know what the different oscillation frequencies are when people talk about alpha, beta, gamma, theta, what they are, but if they actually tie in or not, if you use those frequencies. And I don't think anybody's done that.
Yeah, actually, I think you're right.
Yes.
I wonder if Hanli wanted to comment on that. Hanli, you see your hand?
Yeah, I was going to have some questions, but then to the comment or the question that James is asking, so far by my own experience by reading literature, I'm trying to get my camera on.
There you are.
Yeah. So it's so far nobody yet, or I say very very limited publication that has done that kind of comparison with different, even between CW. I mean, I'm trying to do it, but over 2 years, I still have a hard time to do that kind of comparison between CW versus any kind of modulated frequency, either 10 hertz versus 40 hertz. So, I mean, because my experience, like here too, we particularly with LED, we have to do multiple repeated treatment or intervention, which causes the difficulty to bring them back. And then, of course, if we want to do home-based study, then it gets to the quality control. So that's why, from the community point of view, I think it would be definitely valuable. But so far, I don't know yet, or I have not found anyone did that. I think there was a group who gave the talk back to maybe 2 years ago, a group from China. They did maybe 100 hertz and then 10 hertz and then CW. From their paper, it shows like it's better with modulated signals. I see 3-4 hands up there. So could I ask my questions?
Go ahead.
Then,
sure.
Okay. So I apologize for my delayed join for the meeting. So what the question is, where do you deliver the light? Is it just locally? I think I saw the light on the sort of top. So is that local delivery or is a whole head?
No, it's not a full head. It's four clusters, P4,
Okay.
P8, and one frontal and one in the middle.
And then did you, so it's still a good effort, but then did you test any behavioral change through this 35 5 weeks treatment?
One thing that was observed by the subject is vivid dreams. That was observed, and there was improvement, little bit of improvement subjectively. Objectively, he has done the test, the same test that he was done initially, and it was better. It was better. It was 2 points better, so it was in the, let's say, normal range.
Thank you.
All right, thank you. And Lou, you're next.
Yeah, to answer James' question, actually, my group is very active in exploring pulse frequencies delivered to the brain. And in this audience, I see Jean Chen, who we are collaborating with, and Reza Zomorodi. Now, we published a paper in 2019 showing that when we delivered gamma at 40 hertz, we elevated the power or amplitude of alpha, beta, gamma and reduced delta and theta, which was actually quite a novel piece of finding. And a number of researchers actually latched into that and carried on. We are doing both internal and in collaboration with different pulse frequencies. We haven't yet published a number of them. Reza can add on to this. When we are delivering onto healthy brain, it's actually very consistent that the brain actually responds with a higher power in gamma at 40 hertz. Now, here again, I welcome Jean to talk about the different frequencies we're delivering that her lab is observing, both with fMRI and EEG. That's also pretty interesting. It's not published yet. Maybe some. I think there was a presentation, Jean, that you presented on that too. So all this is very interesting. Now, I think what is going to be very interesting and profound is we are doing the pulse frequencies on high-level meditators, and definitely it has a huge influence in triggering some into a higher, you may say, jhana or bliss state. Very, very interesting. It definitely, pulse frequency definitely has an influence on how the brain's responding. Now, to support that, we are also doing, we published one on 10 hertz on cell culture on the what's happening to tubulins, on its structure as whether it aggregates into microtubules, one way or the other. And again, what was published, 10 hertz actually causes an instability in the secondary structure of the microtubules. And we are in the process of writing another paper and see what the effect of 40 hertz is. And 1000 hertz, they're actually quite different. So in summary, I welcome the others who are collaborating with us to add on to what I say, but definitely pulse frequencies have a big influence on brain response.
Though, if I may, so I think what I'm getting from what you said is that there is some wavelength specificity to the response, as you said, microtubules instability with 10 hertz. However, I guess one of the questions here is how is the syncing? Is there a pacing that occurs for a specific wavelength, an entrainment that is specific to the frequency? And I welcome also Leon next in case. Leon, do you hear us?
Yeah, can you hear me now?
Yeah, we can.
Thank you. Yeah, no, I'm completely agree with Hanley Liu. We also haven't seen in our internal experimentation, particularly with healthy subjects, any so-called entrainment of the signal using pulse stimulation. A lot of that is related with artifacting, corrupting the signal. And more important, we need to evaluate that theta alpha frequencies are maybe the two most variable frequencies on the oscillations. You can change those basically by any type of stimuli, the subject getting bored, falling asleep. So it is interesting always to evaluate the different conditions, if it's at rest, if it's at eyes open, eyes closed, but also under cognitive load. And understand how photobiomodulation works in the neurophysiology. So in that sense, I believe that most of the publication, I believe there is not such entrainment. Most of that is just problem with artifacting, especially how the signal is being delivered. But it will be interesting. I hear a comment, Saša, about the combination of neurofeedback and PBM. At least I know that here in the United States, two groups doing the same type of project research. And one of the main problems they are having is the artifact coming, just like the same happened to you, the artifacting from the source on the main signal. So, I mean, they can filter that and select a wide range of frequencies, but I think at the end, they are deciding to move more into, especially to evaluate the effects going through other type of physiological assessments like ERPs, and not only on spectrum-based analysis. Connectivity is another way to do it. I will do also maybe some, I don't know if you have a thought about entropy analysis, just to see the complexity of the signal and see how much information you can get out of that. But yeah, that's what I believe.
Saša, did you want to comment on that? Otherwise, I see a comment by Jean as well.
Yeah, good idea. Thank you. to look at the entropy of the signals. Yeah.
So I see Jean in the chat says that they found with different pulsation frequencies and different wavelengths, I've found a wavelength frequency interaction in the response amplitude. So this is something completely new that I haven't heard before, that it's not just important the frequency, apparently, according to her data, but also the wavelengths at which the frequency is used. And so very curious to see your data, Jean, at some point. And I see we're getting towards the end. Marty, you had your hand raised. Do you have a comment?
Hi, Paulo, just to respond to your asking for clarification about my question, it was more of a brainstorming kind of thing. I tend to, in designing classifiers, I tend to look to extract as much information from a signal, time frequency features, for example. But to make it a very simple example, my thought was if you demodulate the alpha and theta signal, and you're going to get something as simple as an average frequency after demodulation for alpha and theta. And the question is whether people have, and then the ratio, and whether people have any idea whether post stimulation, let's say an average higher frequency after demodulation in alpha and theta, or the ratio, would be indicative of some sort of optimization of functionality, or a lower frequencies, or if anybody has actually looked at that at all. So,
Saša, you have any comment?
I guess it's more question for all of you. Yeah, as if I understood correctly. Yeah.
Anyone, any comment from the group? Yeah, Marty, I don't have an answer to that. I see also a comment from Harvey. Harvey, I don't know if you want to express what your findings are.
Well, they weren't my findings. It was Ji Shenhong from the People's Republic of China in the 1980s.
I
He
see.
found that different frequencies produce different neurotransmitters. And above 100 hertz, you get enkephalins, which affect the head and the brain. 10 hertz affects the body, and 2 hertz produces dynorphins, which affect the lower part of the body and the legs. 10 hertz produces endorphins. Everybody talks about endorphins in response to acupuncture, but it's not just endorphins. It depends on what part of the body you're treating, what frequency you're using.
Okay. Any other? It seems like Hanli, you're coming back.
Yeah, I am just basically bringing this question to the group because I have a hard time to really get relatively well published or just published references, which is related to mechanism of this EEG signal that are changed by light. So we have been, I think the almost entire community in this field is citing the cytochrome C oxidase, which as the source of the stimulation. But then how to get the stimulation from cytochrome C to electrophysiology, that is something I don't have any reference or anything like that to give something we can cite. I basically read the paper from Lu Lim. He put the paper into Michael Hamblin's edited book. And then he has some meaning. Lu had some hypothesis or anything like that. But so far, I'm really lack of information from references. So if anyone has a good reference, please let me know because that will help us to go say why. And for example, for this presentation, why do we see the increase of power or anything like that? Okay, that's
nodding.
my question.
You know, I mean, my only hint would be to go to the people that are interested in biophotons. I know there's a lot of controversy there, and I see both James and Fred are here, and Liebert, and I wonder if the current mechanisms are somewhat not offering a good explanation for the rapid changes in excitability and brain oscillation, if other alternative mechanism could be explored and see if they fit possibly. But again, that's just a hint, and I'm not necessarily an expert myself. So if anyone wants to comment.
The more I learn, the more confused I am. It never seems to stop. I have more questions than I did 37 years ago when I started.
That's what kindly pointed out, I think, is the central question. If we believe the mechanisms of PBM, how that affect the mitochondrial level will reflect in the neurophysiology of what we are recording. That link is missing. And yeah, a lot of questions. Somebody else needs to answer those.
I'll contact Paolo. I have a colleague who was my student earlier. They just got a one paper published in rats showing electrophysiological changes under, but that's toward epilepsy treatment. So maybe if there is a slot, I can invite actually my colleague to present that.
Yes, please.
But that's for animal. But do you think the animal is relatively okay for this group of presentation?
Yes, we are animals as well, so I guess.
Okay, I'll send his information. Then you can invite him.
Okay. Thank you. Fred, Fred.
That when I use the light clinically or in a clinical trial, we get a clinical response within a few minutes, and with the light. And so I think that there's a mechanism beyond the usual cytochrome C mechanisms. There's something that's happening very quickly. And I think that there are some quantum interactions that are important, and depending on which side you put the light, you get a different personality response, depending on which side you put it on. So I think that somehow the light is interacting with brain information and affecting the subjective response.
Thank you, Fred. Thank you for this last comment, and thank you, Saša, for stimulating this very collective conversation. And thank you all, and to be continued. Take care.
Thank you.
Bye, everybody.